Hello, everybody. This is the audio recording of lecture set number 14, reverse transcriptase HBV, hepatitis B virus. Once we finish this lecture, I'm going to go and do a longer lecture that will cover the entire lecture set number 13. So today we're talking about replication of reverse transcribing viruses, hepatnavirus. So the family that we are discussing today is the family Hepatnaviridae. These viruses cause hepatitis, which is inflammation of the liver. Inflammation of the liver has several etiologies or causes. These include injury, chemicals, drugs, or other infectious agents. The Hepatnaviridae family of viruses are DNA viruses, and they have a tropism for hepatocytes, meaning that they like to infect and set up shop in liver cells. There are the genome structure and mode of replication of Hepatnaviridae family of viruses are unusual because this family of viruses contain partial double-stranded DNA and have DNA polymerase, and they use reverse transcription. These viruses have wide host range. They can be found in humans, animals, and plants. However, they are difficult to grow in cell culture in laboratory settings. The family of Hepatnaviridae has a genus called Hepatnavirus. Hepatnaviruses use humans, animals, and birds as their hosts, so they have pretty wide host range. Viruses in this family include hepatitis B virus, or HBV, woodchuck hepatitis virus, or WHV, and duck hepatitis B virus, or DHBV. Hepatitis B virus is also called human serum hepatitis, and the virus can be found and is transmitted via body and body secretions by close contact. Slide number 4. Hepatitis B virus, or HBV, has an envelope that on the envelope, the 42 nanometer diameter of the envelope, we have 3 surface glycoproteins, the prominent one being hepatitis B surface antigen. The virus also has a spherical virion core that includes nucleocapsid. The diameter is 28 nanometer, and it includes HBC and HBE. Sometimes we only find hepatitis B surface antigen in the blood, so we call this just the glycoprotein in the blood without the capsid or without the nucleocapsid and the genome. We call this incomplete viral forms. These are either spherical or fiber shapes, or they can be elongated and rod shapes. These incomplete viral forms that are found in the blood and are only one of the hepatitis B viral antigens called HBs antigen was initially identified as Australian antigen. Slide number 5. HBV genome, it has a double-stranded DNA genome. The genome is relatively small. It is a circular genome, but not complete and closed circular, because it is partially double-stranded, one of the unusual characteristics of this virus. The full-length negative DNA strand is 3.2 kilobase, and it is generated from reverse transcription of the 3.4 kilobase mRNA, and the partial positive strand is 1.7 and 2.8 kilobases. The genome contains 211 base pair direct repeat sequences, or DR1 and DR2. These are required for DNA replication. And at the end, the genome has terminally redundant ends. Terminally, at the end of the genome, redundant ends due sequences that repeat themselves, so they can circularize, contributing to the circular DNA genome. Moving to slide number 6. HBV genome contains 4 ORF, or open reading frames. There are overlapping genes, but no splicing occurs, and these overlapping genes lead to the translation of 10 proteins in total. The open reading frames are ORF-C, that codes for core proteins I mentioned a couple slides ago. These include hepatitis B, C, and hepatitis B, E proteins. Open reading frame P codes for polymerase proteins. These polymerase proteins are DNA polymerase. It also makes reverse transcriptase and RNase H, and it makes protein primer for RT negative DNA strand synthesis. The third open reading frame is open reading frame S, that codes for surface envelope glycoproteins, hepatitis B surface. Remember, we said that there are 3 surface glycoproteins. These are L, M, and S glycoproteins. And open reading frame X codes for accessory proteins. These are either regulatory proteins or transactivation proteins. Moving to slide number 7, the entry and uncoding of hepatitis B virus. Virus gains entry into the host cells through receptor-mediated endocytosis. So it requires specific receptors on the surface of the host cell. Upon entry into the cytoplasm of the host, we have partial uncoding, and then viral DNA polymerase that is brought in by virus itself completes double-stranded DNA synthesis. Viral newly double-stranded DNA synthesized will enter nucleus, and The free ends of it will ligate to each other. Remember, they have these terminal repeats. And this linear strand will be converted to CCC, covalently closed circular double-stranded DNA. The circular double-stranded DNA will associate with host cell histones. So the mechanism is very similar to episomal DNA, extra chromosomal units of DNA. The messenger RNA transcription of hepatitis B virus. mRNA transcription of HBV is activated by host cell factors. Remember, virus has a tropism for hepatocytes or liver cells. So these host cell factors are in the liver. These proteins will bind to the enhancer region of viral DNA, trigger the expression of 4 partially overlapping, remember we said unspliced, messenger RNAs. This is called nested set. We do not have any splicing of it. These 4 overlapping mRNAs, partially overlapping mRNAs, all have different 5' ends, but they share the same or a common 3' ends, as well as poly A tails. Due to the presence of multiple translation initiation sites on these 4 partially overlapping mRNAs, each viral protein will be translated individually. Moving to slide number 9, the 4 messenger RNAs of HBV. We have 3 different viral promoters located on the DNA genome that host cell RNA polymerase acts on them. RNA polymerase 2 of the host activates viral proteins, leading to the expression of 4 mRNAs with different sizes. They range from 3.4 kilobase transcripts all the way down to 0.7 kilobase transcripts. So the 3.4 kilobase transcript is larger than the genome and is needed for C and P proteins, whereas the 2.4 kilobase transcript codes for hepatitis B surface glycoprotein L. 2.1 kilobase transcript codes for the other 2 viral surface glycoproteins M and S, and the smallest transcript, 0.7 kilobase transcript, codes for X proteins. So how does HBV genome replicate? On the 3-4 kilobase mRNA, there is an internal start site that will be served as a template for DNA synthesis. We have a viral protein primer piece. A protein comes from the virus and acts as a primer that will be used for negative DNA strand synthesis by viral reverse transcriptase. And while we are making this negative DNA strand, the remaining RNA will be degraded by a nuclease called RNase H, and replication goes on until we reach to the end of DNA. And remember, this is double-stranded circular. So when it reached to the end of one piece of this DNA strand, it will jump to the other end to complete the synthesis. This concept of jump, I know I'm doing these last two lectures in reverse order, but we will discuss this jump in a lot more depth and detail when we're talking about HIV for next lecture. So once the DNA synthesis is completed, we will have these terminal repeat sequences that they not only help in circularization of the strand, but they are also important for strand recognition. Remember that for negative DNA strand synthesis, we had a viral protein that was served as primer. However, for the positive DNA strand synthesis, we need viral RNA primer. So for negative DNA strand, viral protein primer. For positive strand, viral RNA primer. Moving to slide number 11. HBV assembly and release. The core viral particles will be formed in the host cell nucleus. Viral DNA genome is inserted before the positive DNA strand synthesis is completed. So what do we have in the genome? We have complete negative DNA strand and partially newly synthesized positive strand. Then assembly will be completed by the production of those proteins required, glycoproteins required for envelope in endoplasmic reticulum and Golgi. And the virus, complete virion, and a small number of incomplete membrane particles. Remember the Australian antigen that we talked about before? This is only HBS, the surface antigen alone in the blood. So both virions and the incomplete forms of the viral particles will be released, and they are found in the bloodstream. Hepatitis B virus and hepatocellular carcinoma. Those people who have an infection of hepatitis B virus have an increased risk of about 200 times for future development of hepatocellular carcinoma. How is this possible? Following viral infection, entry, encoding, replication, etc., HBV DNA will remain persistent as an episome inside of the host cell nucleus, or it gets integrated into host cell. It can either be present as an extra chromosomal segment episome, or will be integrated into host cell DNA. And this process is being modulated by viral X protein. Viral X protein is a transactivator. So activates the transcription of viral genes. And if the DNA remains persistent and we have the transactivator viral X protein present, we have chronic HBV infection while the virus is being continuously replicated. And over time, it results in irreversible liver damage and eventually cancer of the liver or hepatocellular carcinoma. Unfortunately, host is unable to clear this virus by immune mechanisms. The reason for that is we have not been able to detect any antibody against hepatitis B surface antigen in the serum of these individuals. What about hepatitis B vaccine? Is it is a recombinant vector with HBV-S gene. So what we do is when we clone the HBV DNA, that segment of the HBV genome that codes for hepatitis B surface antigen, we will cut it out and put it into a yeast DNA vector. We ligate it so this HBS antigen, this particle, this sequence of HBV DNA will be incorporated into the yeast DNA vector. Then we transform the yeast cells, grow the yeast cells, propagate these cells, then isolate these cells from the culture yeast cells by centrifugation. The yeast cells are heavier, they go to the bottom, then we purify them and lyse the cells, isolate the protein, which is HBVS protein. So the purified HBS antigen forms spherical particles that we use it for vaccination. It is only the envelope glycoprotein without the genome. Therefore, it is not infectious, and it is very safe and relatively negligible side effects. The Center for Disease Control, or CDC, has some recommendations about HPV vaccination that is as following. All newborns should receive a birth dose prior to leaving the hospital, and all children aged 19 and younger should also be vaccinated against HPV. HPV vaccine is the first recombinant DNA vaccine. The concept of recombinant comes from the fact that part of it is the cloned HPV DNA coding for S antigen, and then we put it into yeast DNA vector. So if one piece of DNA from one source is being recombined with another piece of DNA, and this is the first vaccine to successfully prevent human cancer. So the vaccine is called HPV vaccine. The composition of the HPV vaccine is HBS antigen. So the body develops an immunity against it. It doesn't have genome, so it's not infectious. And this recombinant DNA vaccine is the first vaccine that prevents human cancer called hepatocellular carcinoma. Slide number 14 has some information about hepatitis B virus, a little bit more detail about the family itself, as well as the composition of the viral genome. Slide number 15 adds a little bit more onto it. We'll talk about the circular DNA of HPV genome, as well as the covalently closed circular DNAs. For those of you who, like me, may have forgotten about some of these details, I would strongly recommend you to take a look at it. And lastly, we have a summary of all of these steps that we talked, including the attachment, penetration, uncoding, replication, assembly, and release of hepatitis B virus. The life cycle is a little complicated, and another unique thing about hepatitis B is one of the very few known non-retroviral viruses, but still uses RT or reverse transcription as part of the entire process of its DNA replication. So that's another unique thing about it. Slide number 17, the summary. Hepatitis B virus is double-stranded DNA, but it's very unique because it is small and circular and only partially double-stranded. The genome has 4 open reading frames, CPSX, that transcribes 4 messenger RNAs that are partially overlapping. HBV can cause persistent chronic infection that can result in cell activation, meaning that the viral genome will either persist as episome or will get incorporated into host cell genome, and with the aid of the viral X protein, which is a transactivator, activates the cell constitutively, leading to cancer of the liver called hepatocellular carcinoma. The readings for this lecture are from chapter 21, hepatinoviruses, variations on the retrovirus theme. This concludes the audio of lecture on RT hepatitis B virus. Thank you so much for your attention.